Hello everyone, my name's Allie, and I'm going to be sharing with you today the recent research in brain development with regards to synaptogenesis, synaptic pruning, and plasticity. If you have no idea what I'm talking about, that's quite alright, just stick with me. It's important to understand the basics before you dive into any sort of current research. So let's start with the idea that the human brain is like a computer, full of wires and connections. The wires send information to different parts of the computer or different parts of the brain. Unlike a computer, however, the connections in the human brain can be lost if they're not used. Neurons are the wires of the brain. They are specialized cells that transmit nerve impulses. A synapse is the junction between a dendrite and an axon of a neuron. These connections allow for the transmission of information from one neuron to the next. As neurons mature, they extend axons and dendrites. Axons send information and dendrites receive information. By age 2 or 3, the infant has close to 15,000 synapses per neuron, twice that of an average adult. The video you are watching shows the transmission of a chemical message across the synapse. Video provided by YouTube and is from the medical atlas, The Human Brain. Synaptic pruning is the process of eliminating connections that are not being used and strengthening the ones that are. Neurons that are not receiving or sending information may become damaged and die through a process called apoptosis. Synaptogenesis is the process in which synapses are formed. These processes play an integral role in how the human brain functions and how we perceive the world. Scientists have just begun to understand the importance of these processes and look at what happens when one or multiple of these processes fail to occur or potentially go into overdrive. Currently, researchers at UC San Diego have conducted a study on children with autism. They specifically looked at the number of neurons in the prefrontal cortex of the brain. The study involved 7 deceased male children who had been diagnosed with autism, age range from 2 to 16 years old. The male children were then compared to 6 typically developing male children. The study was conducted as a blind study so that the diagnostic status of the children remained unknown to the anatomists. The anatomist counted the number and the size of the neurons in the prefrontal cortex using computerized analysis. The cortical cells were found to be in excess, along with an enlarged head size. Interestingly, cortical cells are not formed after birth. They grow in abundance between the 10th and 20th weeks of pregnancy. During the 3rd trimester of pregnancy and soon after birth in a typically developing child, the excess cells are destroyed. Children with autism had approximately 67% more neurons in the prefrontal cortex than the control group. The weight was also greater in children with autism than in the control group. The cortical cells are what make up 1/3 of the gray matter in the brain. It is involved with social, language, cognitive, communication, and affective functions. These are all functions that are commonly disrupted in children with autism. Although the study is small, and not all children with autism will show similar results, it still begs the question, why are there excess cells? Has synaptogenesis gone into overdrive? Has synaptic pruning failed? Is there excessive neurogenesis, gliogenesis, or a reduction in apoptosis? One theory was posted in the International Handbook of Autism and Pervasive Developmental Disorders was that it could be related to the tumor suppressor genes that are responsible for controlling brain growth. Potentially, they could be defective and causing a failure in synaptic pruning, but that's just one idea. Brain plasticity is another amazing phenomenon of the human brain. Previously, it was believed that if a person had a stroke or suffered a traumatic brain injury, the recovery was minimal and a full recovery was unthought of. But as you can see in front of you is Gabrielle Giffords, who suffered a traumatic brain injury and has started to recover. That is what they believe to be brain plasticity. Brain plasticity is the idea that the brain is always changing and adjusting. Neuroplasticity is not a single process, but rather it's several processes that occur over a person's lifetime. The brain makes changes in response to experience and sensory input. Neuroplasticity occurs in 2 primary conditions. According to the principal activities of brains are making changes in themselves, according to Marvin L. Minsky from the Society of Mind, 1986. First, during normal brain development, when the immature brain first begins to process sensory information through adulthood. Second, as an adaptive mechanism to compensate for the lost function and/or due to brain injury. When an injury is sustained or a person suffers a stroke, brain cells are often damaged beyond repair and eventually die. Regenerative processes are activated and the surviving neurons sprout nuclei and axons. New neurons are also produced, which form new synapses. Originally, it was thought that new neurons could not be produced. By knowing that the brain can recover from injury, therapists can understand how to create therapies that will continue to help the patient regain lost or damaged functions. Different therapies that are beneficial are physical therapy, occupational therapy, and speech therapy, to name a few. It's important for patients to use whatever it is that they have lost, whether it be their arm or the ability to speak, because if they don't, the chances of recovery greatly decrease. If you don't use it, you lose it, according to occupational therapist Stephanie Brainard. It is important to note that although recovery is possible, the timing and location of the damage may greatly increase or reduce the chances and amount of recovery. The brain continues to be a puzzle which scientists seek to understand. Luckily, we have new technology available to help us get a better image of the brain. Such technology includes the MRI, the functional MRI, the sleep MRI, the diffusion tensor imaging, the structural MRI, the MEG, and the PET. Scientists are able to open a window into the human brain and the developing brain. Although the study done by UCSD was small and only included males, I think it is still going to open a whole new area of study with regards to autism research and diagnosis. The new brain research is going to shape future ideas for therapy, for brain injuries and strokes. It's going to change the medical world as professionals and patients alike learn that the brain can recover. Thank you for sticking with me, and keep an eye out for more research on these topics and others on the mechanics of the human brain.